The overall goal of this procedure is to encapsulate small pockets of liquid between graphene sheets for imaging nanomaterials using transmission electron microscopy, or TEM.
This method can help answer key questions in the nanomaterials field, such as the growth and etching of nanocrystals, with the high spatial resolution of electron microscopy. The main advantage of this technique is the ability for research groups to perform liquid cell TEM experiments using an existing holder with low startup costs and high spatial resolution.
Generally, individuals new to this method will struggle because handling graphene-coated TEM grids is difficult and requires a lot of practice.
To begin, cut out a roughly 2 square centimeter piece of pre-made graphene on copper, which fits around 6 to 8 TEM grids. After cleaning the graphene as detailed in the text protocol, smooth out the graphene on copper piece to remove any macroscopic wrinkles. To do so, take 2 clean glass slides and place a folded wipe on the bottom glass slide. On top of the wipe, place the graphene on copper piece. Finally, place the second glass slide on top. Press down on the top slide, gradually smoothing out any wrinkles in the graphene on copper piece. Reduce the number of folds in the tissue and repeat the pressing process. Continue the process until a final pressing between the 2 glass slides with no tissue wipe. Lay down the TEM grids on the graphene on copper piece by first placing the wholly amorphous carbon support foil TEM grids down on the graphene with the amorphous carbon in contact with the graphene. Place a couple droplets of isopropanol on the grids.